Hello doctors, myself Dr. Switra, Assistant Professor of Biochemistry. Welcome to the Raaonline. I'm here to take biochemistry sessions. Today we'll start with the session on cell and cell organelles, and this is the curriculum we are doing based on competency based medical education. So we'll look into the session today. What is actually a cell? Cell is a structural, smallest functional unit of life. This is the basic structural and functional unit of our life. The cell, it is the building blocks of life. What do you mean by the building blocks of life? Because this cell, this smallest unit, it is built upon each other to form a tissue. These tissues, this is a tissue. And these tissues are organized to form the human organs. So, all the human organs are organized by the tissues, and the tissues are organized by the cell. So, the cell is very, very important to our life. So, how the cell is studied? The cell is studied by fractionation, by differential centrifugation. The cell is ruptured and the homogenate inside the cell is studied. So, tissue cells are ruptured by extracting in a mild condition. Why? Because the cell organelle activity should be intact, so that they are extracted in a mild condition. Then it is put in a mixture like thing. So, it forms a homogenate. It undergoes homogenization and it forms a homogenate. This homogenate contains cellular organelles. So, how the cellular organelles are separated? By this differential centrifugation. So, at various speed, like I show in this slide. So, here at various speed, see, this is 800 times the gravity, here 15,000 times the gravity. So, as the speed increases, various organelles are separated, like nuclei, mitochondria, lysosomes, peroxisomes, and all other organelles are separated when the centrifugation is done at various speeds. But how will you confirm the purity of the cellular organelles? By marker enzymes. What is marker enzymes? So, the marker enzymes means these are the enzymes. They are specific to the target cell organelles. So, that cell organelles contains that specific enzymes. They are called as marker enzymes. When you separate the cell organelles, the marker enzymes are released. These marker enzymes, they prove the purity of the cell fractionation. For example, if this is the cell, the nucleus, this is the nucleus. So, this contains polymerase enzymes. So, polymerase means DNA, it may be either DNA polymerase or RNA polymerase, because the nucleus function is helping for transcription and translation. So, this polymerase are present in the nucleus alone. So, they are specific to the nucleus, and so it is called as marker enzymes for nucleus. Next, we'll see about the subcellular organelles. First subcellular organelles which we look into is nucleus. So, this is the nucleus part of the cell. So, nucleus is also surrounded by membranes. Nucleus contains an outer membrane and an inner membrane. This outer membrane is continuous with the endoplasmic reticulum, and the inner membrane contains so many nuclear pores, and this is called as nuclear lamins. Nuclear lamins provide support to the nucleus. They give the structural support to the nucleus. Without that, the nucleus will be damaged. So, the outer membrane is continuous with the endoplasmic reticulum, and the inner membrane contains nuclear pores. What is the importance of nuclear pores? They allow only selective materials, selective ions or solutes to outside and inside of the nucleus. because this is 100 nanometers in diameter, whereas it allows only 9 nanometer diameter substances inside and outside of the cell. This is because the nucleus content should not leak out of the nucleus membrane. To protect the nucleus contents inside, this are arranged, these are made like that. So they are 100 nanometer in diameter but allows only 9 nanometer dia substances. Next comes Nucleolus. This is the dense material found in the nucleus, and also the chromatins. The chromatins contains the DNA genetic information. That is why the nucleus is called as the storehouse of information. They store all the informations of your body, of your cell into this DNA. It is highly condensely packed along with histones. This is a DNA packed with histones. Okay, so this is the structure of the nucleus.